Alrighty, here we go. So good morning, everyone. Hope everybody's having a good day. And oh, that's not the right one, is it? Let's make sure I share the right one. Yes, this one right here. All right, let's see if I can do this. There, okay, that'll work that way. Trying to learn new tricks. Okay, so picking up where we kind of left off, I'll note that this is, I saw over the weekend, or maybe it was yesterday, that a lot of people in lab hadn't done the antibodies lab yet. So after today's lecture, should be well prepared, somewhat prepared, at least familiar enough that it should go through there. So let's make sure we get that done by Thursday. We're talking about, again, our proteins interacting with their various ligands. So we're going to look at antibodies and antigens as an interaction as a type of this as well. Okay. Yes.
I seen posting on the lab. Do we do that one as well before Thursday?
No, that one will be the one that will show up on. I'm just trying to get them out there ahead of time. Although the good thing about those labs is that you can kind of do them as they become available. So it's up to you, like when you have the time frame. So if you want to do like 2 or 3 at a time. I'm trying not to post them all with due dates right now, but they'll all. I may end up doing that just because I forget to put the due dates in there a lot. So everything will have due dates now, but it'll be like basically 1 a week through up until we get to November. So, yeah. All right. So if we look at our antibodies and our antigens in here, we can talk about the immune system, okay? And we really have kind of like 2 types of immune systems circulating around our bodies. We got the cellular immune system, which will target the own cells, that anything that's been infected that's expressing, say, bacteria or viral infectors, viral infections, or vectors. Choose one. It'll clear up virus particles and infecting bacteria, target the own cells that are bad. Question? Okay, we're good. Okay. And the key players in there are going to be our macrophages, our killer T cells, and our inflammatory T cells. Always, because I grew up watching Arnold Schwarzenegger as a Terminator, and so they're Terminator cells. No, okay. But macrophages are of particular interest because we can actually have, differentiation of different types of macrophages. And we've got M1 and M2, and I believe M1 macrophages are the kind of like, oh my God, there's so much damage, just kill everything. And it's like the walls collapse, just throw up some boards and something like that to fix it as fast as possible. Whereas the other type, which I believe are the M2, are a lot more precise and clean up a little more slowly to rebuild. So M1 might lead to more scar tissue, M2 might lead to more regeneration. So that might be a topic in medicine where you can figure out how to get those moving through there. All right, let's see if this is working the way that I want it to. Okay, good. Random emails. Okay. All right. We also have the humoral or fluid immune system. This is going to look at extracellular pathogens. These will be things like, say, in the extracellular matrix, if you have bad stuff there, possibly damage, possibly foreign proteins, but we make a lot of soluble antibodies. The other thing is, this is kind of neat because our fluid immune system keeps a memory of past infections. So if you look at your helper T cells and your B lymphocytes, they're the ones they're going to remember. And it's kind of like if you get chicken pox, you're not going to get it again because you're going to have this memory of antibodies that keeps circulating as well, and looks for those particles so that it'll prevent a full-blown infection if they come back again. All right. Let's take a quick look at our cellular immune system here. Our antibodies are going to bind to fragments that are displayed on the surface of invading cells. So in this case here, we have a viral infection agent, a viral vector. Our antibodies are going to bind to the surface of this antibody. They're going to recognize some of those proteins and say, hey, this shouldn't be here. So the antibodies bind to it. And then the other end of the antibodies will bind to receptors on the macrophages, which will induce the macrophage to endocytosis, bring it inside, and basically kind of chew it up. Okay, so yeah, you get into a lot of interesting things with macrophages and what they can do, and then immune responses, immune diseases. You can then end up with plaque formation if your macrophages don't end up dissolving or something happens with them, but that's beyond what we want to do. I just find them kind of interesting, so maybe we'll do a case study on those later. All right. Our humoral immune system is the one that we're going to talk about a little bit more, because now we're looking at the memory. How do we know? How does our immune system continue this response? So we have these soluble antibodies that kind of go through the bloodstream, go through the ECM, and they specifically bind our antigens. So those are things that that the body says shouldn't be there. This is why a lot of autoimmune diseases are particularly insidious, because the immune system is recognizing something that should be there as something that shouldn't be. So you're essentially attacking your own body. However, they're designed to attack things that shouldn't be there. Typically, these are going to be macromolecules, maybe long stretches in here. These will often be the coding proteins that you find in the bacterial cell wall or on the capsid of viri. And you can also recognize carbohydrate chains as well too, because again, keep in mind our cell wall is going to be in particular heavily glycosylated. Same thing with our coding proteins of viruses often have big stretches, large amounts of carbohydrates that are part of that outer surface. So our antigens, our antibodies will recognize these particular antigens, these particular sequences, and bind to it. So the antibodies are the good thing, and antigens bad thing, antibodies good thing. That's our simple. Go back and do some old 90s improv here. But the antibodies are proteins that specifically recognize those antigens. They bind to it, and when they bind to it, it's essentially a marker. It's like, hey, I found one, over here. So they're going to mark it for destruction when it binds there. So it binds to a very small region. The region that it recognizes is known as the epitope. Okay, so one antigen, say you have like a macromolecule, a long carbohydrate chain, maybe it's 1,000 molecular weight. It might have a sequence of 20 or 30 molecules in there. So you can recognize, typically our antibodies are going to recognize a 4 to 8 sequence section. So we could have multiple epitopes on there. All right. Pictorially, the way this works, we draw our antibodies as our kind of our Y shape. You got your light chains and your heavy chains, because they're longer, so they're heavier. You have your antigen binding site up here. This is our variable region, all right? And then the C's are our constant region, okay? So the variable regions are the ones that, each little section there is slightly different, and that's how it determines what it will bind to, what its specific binding partner is, or another way of saying that, the particular antigen that it will recognize, okay? So this recognizes, a variable region recognizes a very specific sequence, and if it finds that sequence, it will bind to it, and then the other end here will say, hey, come here, we found one, got one, got him. All right, and then this variable sequence between different antibodies is variable. So we might have antibodies that recognize the same antigen, but different epitopes. You would expect that this might be fairly similar, but it's just recognizing a different site on there. If we look at our antibody in a three-dimensional structure, we can look at immunoglobulin G or IgG. All right. My little aside for the Mandalorian coming up here. Well, even though it's not really, it's IG-11 in there, but when you're making a Star Wars reference, you got immunoglobulin A, right? So I need to find a version of the T cell in there, and I can call it IgA T8. Ha ha ha. But anyway, this is our three-dimensional structure, and we got our variable region up here. I recognize it. Brown carbohydrate in here for our activation. And again, we're going to see induced fit. So if we look at our particular binding region in here, there's no antigen bound. Here we have it with our antigen bound. And it's not easy to see, but we have some slight shifts in here. Okay, they don't show the antigen here, but we're just looking at how this sequence changes, so our three-dimensional shape. And that's not terribly easy to see. In fact, I'd kind of expect you to kind of look at this and go, that really looks very similar. But other than like this is kind of squished down a little bit, and this one's kind of pushed out a little bit. Maybe this section here looks really similar, right? But now we've got this little finger that kind of extends down here. So these are very subtle changes, and I'm not going to expect you to delineate or talk about those changes there. But the important part is with these subtle changes, that's what's going to trigger the downstream effects. So if we find something, we have this induced fit. That induced fit induces a conformational change. The conformational change will activate the next step. It's like you'll turn on the light that says, hey, come get one. So rather than actually try and interpret little 3D structures like this, it's a lot easier to draw antibody and antigen interactions as cartoons. Okay. So this is our big payoff slide here. Our antibodies are, we can use antibodies in a huge number of ways. So besides the cellular defense, if we have antibodies, we can detect very small quantities. Because of the high specificity of antibody-antigen interaction, we have extreme specificity. That means basically if we have a way of detecting the antibody, we can detect very small quantities of our target protein, of our target antigen. Okay? So that's what we do. So we may have a cell surface, or some surface, maybe a petri dish. We coat it with a variety of sample in there, okay? And we look for particular antigens, okay? We're gonna block unoccupied sites with a blocking agent. If you've ever done maybe in a biolab, pour some milk over your sample, the casein proteins are functioning as the blocking agent in there, okay? And once you block all those sites, we do that so we don't get non-specific binding. We wanna incubate with our primary antibody. So our primary antibody is going to be the one that recognizes our antigen. Okay. So if our antigen is there, in this case, the little blue one, our primary antibody will bind to it. So then we'll wash everything off, and then we'll treat with a secondary antibody. Okay. And our secondary antibody recognizes our primary antibody. Okay. And the reason we're doing this is for amplification. So we draw just one on here, but we actually might have 3 or 4 or 5 secondary antibodies binding to our primary antibody. All right. At the tail end of our secondary antibody, we have a signaling molecule of some type. This might be a horseradish peroxidase conjugate. It might be an alkaline phosphatase conjugate. Something could be radioactive, but we don't do that. But typically HRP or AP are the 2 standards that are in there. So once we've treated that and rinsed, we can add substrate here, and then we'll get a detectable color change. Okay. Sometimes this might be fluorescent. We might need to develop this on, say, like photofilm, or hit it with laser to stimulate it or something like that. But the simplest ones will just see a color change. Okay. So in this case, the yellower we get here, I think this is an HRP. This might be alkaline phosphatase. Not that it really matters too much, but yeah, this is horseradish peroxidase. Okay, so basically the more protein, the more antigen that's there. So the more primary antibody that's bound, the more secondary antibodies bound to the primary antibody, the deeper the color will get. So we'd say, hey, we have a fairly high concentration over here, maybe a low concentration here, and nothing in the ones that are clear. Okay, all right. So let me jump back up to this for a second, because how do we actually do this? And our primary antibodies oftentimes are going to be monoclonal, right? So I think this is in the text, but it's not in the slides. So yay, it's like 95% of the stuff is in the slides, but not everything. Our monoclonal antibodies only recognize one particular epitope. So we might have, say maybe a protein that has like 20 possible binding sites. You might have polyclonal antibodies that'll recognize all 20 of those sites, right? But our monoclonal antibodies will only recognize one particular site. And you can grow those up through specific cloning. You pick a particular clone for an immune response and develop your primary antibodies. Naturally, they tend to be a little bit more expensive because they're much more specific. Okay? Polyclonal, you don't need to focus on one thing. Sometimes polyclonal antibodies are good enough. If we would just want to see if a particular virus type is present in some way, shape, or form, maybe a polyclonal antibody will work there. But monoclonal antibodies will allow us an incredible level specificity. You can have a monoclonal antibody that'll distinguish between the exact same sequence if it's phosphorylated or if it's unphosphorylated. All right, so just changing a phosphate group, like 3 or 4 atoms, you can detect that difference with a monoclonal antibody. Okay, so our primary antibodies are most often monoclonal and recognize one particular epitope. Our secondary antibodies, which recognize the primary, are often polyclonal, and they'll recognize several epitopes of this, Y chain here. So you could actually have multiple secondary antibodies binding to that first one. Okay? The way we do this is maybe we'll raise our primary antibody in rabbit and our secondary antibody in goat to recognize rabbit. So it'll be polyclonal and it'll recognize any kind of rabbit particular DNA that's down here. And you'll have lots of them bind. Another reason that's helpful is the more secondary antibody you have bound, the bigger the color change you're going to see, the easier it is to see the difference in there. Technically, we really don't need secondary antibody. If we attached, say, horseradish peroxidase to the primary, you could get a color change in there, but we just might not be able to see it. We do the secondary antibody for amplification of the signal. Okay, so we're going to see a much bigger jump in there. If we go all the way back to, what was that? Chapter 2, I believe, where we talked about amplification as one of the ways. That's what we need here, amplification. And we'll see that again in like chapter 12, I believe. Okay, so the ELISA assay is a very common biochemical assay. I think we will actually be doing that in a simulation a little bit later on in the semester as well too, where we use our antibodies to determine whether we have any of this protein here or not. Okay. So it's just a good way to detect stuff. So that's all I want to say about antibodies. That's a lot on that previous slide. So what is that, slide number 51? There's a lot going on there, so definitely make sure probably see a question coming there, about the use of antibodies and specificity and monoclonal versus polyclonal and all that good stuff. All right. Okay. Questions about antibodies? Stop there for a second. Not too bad, I don't think. And I think generally we get pretty good experience with antibodies in a couple different undergrad labs. All right. So let's look at our third case study for our proteins interacting with ligands. And we'll, that's what I forgot to load up. All right, I'll find it. But we can have muscle proteins, muscle structure in here. All right. So we want to think about that because you think about muscles, just allow you to move. But how does it actually work? It relies on the actin-myosin interaction and a binding and release of some substrates to allow your muscles to contract and relax. So if we look at a structure of your muscle, you basically have a bunch of muscle fibers in there. So if we look at these, you got your capillaries to bring oxygen and take away CO2. You'll have nuclei in these cells as well too. And you'll have this little muscle fiber in here. In the fibers, you have these nice little bands, sarcomeres that are in there to make these myofibrils, and then they'll line up. Basically, the different discs are relative density in there. So we're essentially kind of weaving stuff together. What we have here are some myofibrils in here. All right. So we have this kind of 2 chains that are wrapped around each other. Again, we have light chains that are associated in here and heavier chains on the outside of here. So we have these little head groups that are relatively short, about 10% of the length of the overall fiber. And then we can just kind of weave these together. So our little head groups are kind of holding them together here to make a myosin fibril, filament, sorry, filament. Gotta get my terminology correct here. And then you can see this is about 2.5 times as long. All right, so what do these do? All right, so our myofibrils contain thin filaments of actin. All right, so this is our F-actin here. We have our G-actin subunits. We put them all together and we get F-actin. Always makes me think of the old commercials like tough actin and actin for athlete's foot. So you need your, I don't know, if you're gonna be an athlete, you gotta have actin so you can have your muscles moving. That's terrible. Anyway, the myosin thick filaments slide along those actin thin filaments, okay? And that's how our muscles contract and relax, because they basically slide along there. So they're going to interact down here, all right, to allow contraction and expansion, okay? So if we look at these filaments that are sliding along here, again, we can see for contraction, we kind of pull those together. For relaxation, we let them slide apart. Okay, so here's our relaxed form, here's our contracted form. Okay. So how does this work? Okay. This is going to be modulated by chemical energy. We're going to use ATP. So whenever you do something, we need energy, right? Got to have a power bar or whatever. My energy drink. My energy drink is Mountain Dew. It's terrible. All right. But we're going to use ATP, our adenosine triphosphate, and induce a conformational change in the proteins to allow them to move, to do what they need to do to get that contraction and relaxation. All right. We're going to call this essentially a type of motor protein. Okay? Our motor proteins are just like your motor in your car driving along. We're going to drive stuff along here. So we're going to have these conformational changes that occur in the actin-myosin cycle. All right. So if we look at this on this next slide, we have an awesome picture of the actin-myosin cycle. We're going to start here. Here's our myosin head group. It's interacting with our actin filament. It's kind of bound there. All right. What's going to happen is our myosin head group has a binding site for ATP, so ATP is going to bind there. All right. When ATP binds, we're going to see that the myosin fibril, that head group, can hydrolyze it. So we're going to hydrolyze ATP and get ADP, an inorganic phosphate. As that hydrolysis occurs, our head group is going to move. So we're going to see that conformational change. We have this bound, have this move. When that moves, all right, it's going to cause the myosin to interact with a different section of the actin filament where it can bind there, all right, as we release the inorganic phosphate. Okay. All right, so that binds there, and then we can release our ADP, and as that happens, we slide back to that original position. But now it's bound to the myosin, so we're actually moving. So, and then we can just repeat this. Okay. All right, I'm going to come back to this in just a second, but what I want to do is, let's see if I can pull this up here. Yes, all right, oh my goodness. Okay. What I meant to do, all right. Why are you not letting me type in here? Huh? See if we can see this. All right, so we have this lovely little picture. Yes, maybe. All right, so this is, yeah, you can get this terrible thing in the middle here, right? All right, so essentially, we're just walking along there. So here's our step, our little power stroke, all right? And although this is a cartoon, you can actually use some pretty advanced imaging and kind of see this movement going on there. All right, so let's run through that one more time. But again, you know, that 4-step procedure, you know, we've got our ATP binding, we release, we flex, we bind, we release and flex back. So we can essentially get this walking along here. And in this case, we're carrying something along an actin filament. So we're actually kind of transporting something through there. So that's essentially how that works. Okay, so now No, 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 no. All right, so now I can get back to here. Yes. All right. I know there's a tabbing procedure, but apparently I didn't learn it. But again, that whole walking step, so even though that's really fast there, we should play that like 1/4 speed and say, oh, okay, so it's bound. Oh, ATP comes in and it unbinds. We hydrolyze ATP, we shift position, we release phosphate, we bind to a new position there, we release ADP, and we flex back to the original. So we've kind of pulled this along, okay? And then we repeat this whole step again. So now we can basically walk along the actin filament. For our muscles, we're really just kind of walking along and pulling back and then relaxing to go back the other way. But you can actually, you know, have motor proteins that carry stuff someplace as well too. Okay. So knowing the actin-myosin cycle is probably a good thing. All right. Just some extra information that you probably won't need to know as much is we can regulate this as well too. So our peeking ahead into second semester, everything's about regulation. Yeah. So we can regulate this. This is basically to prevent muscle spasms, continual contraction. I don't know if you've ever like worked out and you get like a little spasm in your arm or your some muscle or something like that. But you want to avoid that continuous contraction in there. So our nerve impulse is going to release calcium. We're going to see calcium plays a huge role in a lot of signaling. But that calcium release is going to cause a conformational change to this tropomyosin troponin binding site. All right. So that will expose these myosin binding sites and allow our troponins to either bind or kind of come off, which can inhibit or promote muscle contraction. All I really want to say is just kind of a peek ahead. But we basically have multiple sites within this single protein that are all playing specific little individual roles to help regulate the total protein function. Okay? So we can make it complex, but we're trying not to right now. All right, that's everything in chapter 5, right? It's like, that's a kind of a lot, but you know, our main focus was on hemoglobin, myoglobin. See lots on there. You know, maybe a question on how antibodies, antigens work. Maybe a question on the actin-myosin section in there. But really, we're kind of looking at this binding, okay? All right. Deep breath. You guys ready?
It's 4, 5, and 6 on it?
Yes, yeah. So I'm thinking not this coming Tuesday, but the following Tuesday. I think that's like the 6th or 7th of October or something like that. Yeah. Oops, there we go, come on. Yeah, and I'll post that. So we've got some time. All right. No. Yes, we will. We will definitely do one. Did I close the whole thing? All right. Nope. This is what I want. Okay. All right, so now I need to go back to Zoom. All right. So, you know, 4, 5, and 6 is kind of a lot, but 6 and 5 really, you know, this is all proteins. They all tie together nicely, all right? So a lot of what we're going to see at the start of this chapter is just going to be a very specific case of protein-ligand interaction, okay? So in this case, you know, we're going to be a little more focused on you know, maybe if we say like our protein ligand is our generic, you know, like Dr. Thunder. I don't know if you've ever seen that generic cola. Instead of like Dr. Pepper, you know, you get Dr. Thunder or Big K Cola or something like that. This is going to be our name brand here. Okay, so our enzymes, we're going to learn about what they do and why they're important, and then we're going to look at some descriptions of how they work, and then the part that everybody loves, kinetics, which is funny because I actually really do like kinetics. I thought kinetics was a lot more fun than some of the other qualitative stuff. All right, so what are enzymes? They're basically biomolecules that have catalytic power, okay? They're going to induce a reaction to occur, okay? So they'll increase reaction rates without being used up, We can put a little star here. If you go back to like organic chemistry, it's like, oh, our magnesium in here is a catalyst. It just enhances the reaction. You know, without interacting, we'll often, covalently change our enzymes, and it might take 2 or more steps for them to change back. So they may specifically participate in the reaction and donate groups and undergo, covalent modification, but eventually they'll be regenerated back to that original form and can do it again. Okay? So our enzymes are catalytic molecules. Most of them are globular proteins. We do have some RNA ribozymes. Ribosomal RNA can catalyze reactions as well too. Again, leading some evidence to the idea that RNA was the first, biomolecule of living organisms to develop, because it could both store information and catalyze reactions. And then we became more specialized. So enzymatic processes date back to the 1700s. They don't really tell you too much about this, right? Some of our earliest biochemistry was like, how can we make beer? Better beer, make beer, So then we also get into textiles as well too, a little bit. But so it's relatively young in terms of science, but still a couple hundred years in there. Enzymes are some of the biggest, fields of study in biochemistry, because they just do so much. And of course, we could have like, old people, there. Yeah, I was gonna say, hey, this name sounds familiar, right? Haldane, isn't there like an Buchner? He probably used his funnel. Okay. Right, yeah. All right. So why do we have biocatalysis over inorganic catalysis? If you think back to organic chemistry lab, yeah, it's probably like put in strong acid, right? You got to do a lot of stuff in the hood and then like boil the crap out of it, crank up the flame, right? That's not conducive to living organisms. We don't want to have really high acidic conditions. So our biocatalysis, we have relatively mild ones. We can, have this work at a pH of 7 instead of like pH of 1, okay? We can also do this at, say, 37 degrees instead of 400 degrees. So it's milder, and that's going to be similar to the conditions we're going to find in our cells, okay? The other thing is, too, if you got like a 50% yield in organic chemistry, that was often pretty good, right? It's like, I talk with Dr. Kieran sometimes, too, and it's like, oh, 17%. It's like, yay, we're up to 17%. I'm like, whoa. So what we see in our biocatalysis, it's 100%. Or if things go wrong, it's 99%. So we make one product and one product only. So it's hyperspecialization there too. So you don't have to worry about, shunting and making minor products or, other unintended, unuseful. So biocatalysis is more efficient. Typically, we're going to see higher reaction rates that occur for a biologically useful time frame. Right? And the other neat thing about them is because we have these big molecules, they have sites for regulation. So we can turn them on and turn them off, or slow them down and speed them up. All right, so if you look at saying something like this particular compound that we have here, in a reaction, you might get 4 different products. Our cores mate mutase is going to give you only this product. And notice we have stereospecificity here, all right? So we can make a very specific, including even the 3-dimensional structure and RRS values for it. So we're going to make one desirable molecule, which can then participate in whatever reaction, however we need it. Okay. All right. Let's mention briefly selectivity. All right, this is a nice advantage here. If we have phenylalanine hydroxylase, we can bind phenylalanine and we can hydroxylate it. Okay, so there we go. That's what we make. If I put in the wrong orientation, here we have L phenylalanine. If I put in D phenylalanine, it doesn't even bind because it doesn't fit in the binding pocket, right? So I'm only going to interact with the ones that'll fit. And in here, I would have an example of something that might be an inhibitor. It has a structure that's very similar to phenylalanine, so it fits in the binding cleft, but in part because it already has a hydroxyl group there, it's not going to be hydroxylated, so it binds, but there's no reaction. So we'll see later on, this lower molecule would function as an inhibitor because it'll basically slow down the overall enzymatic rate by by preventing that enzyme from working on the phenylalanine. Yes? Because it's the wrong conformation. So this is our L conformation, and this is our D conformation. Because it's in the wrong shape, you know, we have a wrong, yeah. So even if you rotate that around, so that it's in this way, our CO2 group is going to be here, and our NH3 group is going to be over here, and the enzyme won't permit that to fit in the binding cleft. Okay. If we look at an example here we have chymotrypsin with a bound substrate. So our key active site amino acid residues are going to be interacting with our substrate. That helps with the specificity. So we have to have this very, very specific shape and size and charge and everything in there for our substrate to fit. Okay. And again, it's going to be an induced fit, so it's not a perfect fit, but it's going to be enough to be very specific. All right, I'm going to scare you guys. Here's our first scary slide. Don't worry, we're not going to do calculations like this here. We could, that would be fun, but most students don't find it too much fun. So our enzymes don't affect the equilibrium, all right? So they don't affect KAQ, which means that the enzymes can't affect the overall free energy of the reaction. So slow reactions have this huge activation barrier, all right? So our enzymes are gonna increase the reaction rate by lowering that activation energy barrier, okay? So we could actually kind of plug this in this way. You can see, you know, our Boltzmann's constant, our temperature, and all this stuff in here. So the way this works, we don't affect the equilibrium of the binding. We do affect the activation energy, our delta G double dagger, okay? So we can see that some reactions are greatly enhanced, okay? Like cyclophilin, you know, we can see that this is, you know, 100,000 times faster with our enzyme. We go all the way down here to our aridine monophosphate decarboxylase, which is 10 to the 17th times faster. So essentially, in the history of the universe, maybe one molecule of this would spontaneously be decarboxylated, all right? But this is a reaction that occurs in your body. This is occurring, you know, was it 100 million billion times faster than an uncatalyzed reaction. Same way with like urease in here as well too. That's ridiculously catalyzed. So the way this works, we can look at our reaction coordinate diagram. This is for our uncatalyzed reaction. All right, here's our energy barrier here. Okay, so our overall delta G is slightly negative, slightly favorable. We just have this huge barrier here. All right, so if we look at you know, our product to substrate, we see our delta G in there. The way our catalyst works is we lower that big old speed bump into like a rumble strip. Okay, so this is really specific here. We normally, if you go back to organic chemistry, you'd say your catalyst, you'd have just like a smaller bump there, right? Okay, but what we're actually doing here is we're ending up making 3 little bumps, okay? And the first case is, here's our enzyme binding the substrate, okay? So it takes a little energy to get the substrate in there. And then once the substrate is bound, that's fairly stable. Then we have our actual reaction that occurs. So our substrate gets converted to product. That's our middle one here. And then our last one is releasing the product and getting our enzyme back free so it can bind more substrate. So that's why we have 3 peaks here instead of just 1, okay? And again, this is our delta G double dagger of the catalyzed reaction, much lower. And typically, our substrate to product is still going to be the highest one here. If I were drawing this, I'd make this, you know, just a little bit lower, so you can see that that central peak was the higher of the 3, which is the way it typically is, not always, but typically. Okay. Does this diagram make sense? So, because we're going to talk about what happens as substrate binds. So keep in mind, we got a couple things that can happen. My substrate can bind, and it can fall off with nothing happening, so it can leave. So it'd be like, you know, some person could wander down the hallway, come in here and sit down and say, hey, I wonder what this class is about. And then they could see this diagram and get up and say, nope, not for me, and leave. Or, you know, be like you guys and come in here and sit down and presumably be catalyzed and have a greater knowledge of biochemistry as you go through there. So there's a couple different things that can happen. You don't have to be converted to product. You can fall off and stay substrate. Okay. All right, so how does this actually work? All right, we're putting things in the proper orientation, and I've got a nice picture coming up on the next slide to show this. So if we kind of talk about the background theory here, an uncatalyzed bimolecular reaction, you got to have two molecules and they have to hit each other in the right way, right speed, right energy, right conformation. And if they have all this freedom of rotation, that's pretty rare. It's going to be statistically improbable. Okay. All right. If we have a unimolecular reaction, now maybe I have one big molecule here, and my arms are connected to the central trunk here, so they're flailing around, and maybe they'll hit in the right direction to form a reaction. So that's a little more favorable, because those, instead of having two molecules that could be anywhere in the room, I now have them constrained so they're relatively close together. For the catalyzed reaction, what I'm doing is I'm physically holding these two reactants close together in the right orientation, so it's very likely they'll react. So I basically kind of force them together here, like so, and they're just close enough to grab on. Okay. All right. So pictorially, we look at this. Here we have, in reaction A, we have these two free molecules. What is this, acetate, I believe? Yeah, so some type of ester with a carboxylate group to form an anhydride. So if we have the two of these together, we might call this a rate of 1. Just arbitrarily, we're going to call when these two molecules get together and form our anhydride, we'll call that whatever that rate is 1. If we actually connect these with another little chain here, we still have some freedom of rotation about these bonds, but these are physically constrained, so they're closer together, so they're more likely to react. So this is going to be 100,000 times faster than if these two molecules are just freely floating all over the place. If we build another bridge here, so now we've eliminated the freedom of rotation around here, and we've greatly constrained this because we have a steric group that's interfering up here. Again, we see that these are now much more likely to interact with each other. And we're going to see that rate is going to be 10 million, 100 million times faster than if it's a bimolecular reaction. Okay. So essentially, what our catalysts are doing, our enzymes are holding our substrates in the proper orientation to make them more likely to react. Okay. I always like analogies, so I'm going to try and make a really bad analogy here. It's like, what was it? You guys remember some shows like one of those dating shows, like Millionaire Matchmakers? I don't know. I remember it vaguely being across there. So it's like, oh, it's so hard to find someone to date. So there's a dating service and kind of puts them together in the right orientation in hopes that they get together. Okay, so that's essentially what our enzymes are doing. Our enzymes are millionaire matchmakers. Ugh, that's terrible. Probably the worse the analogy, the better it'll stick in your head. So, right? At least that's the way it is with me. Okay. So again, we're trying to lower our delta G. That should be a double dagger and not an equal sign in there. The enzymes will bind to the transition states best. Okay, so this comes from Linus Pauling. You probably actually read about more in physics than in biochemistry, but same type of deal here. The enzyme active sites are complementary to the transition state of the reaction. They're not complementary to the substrate. They're not complementary to the product. They're complementary to that transition state. So as they get closer together, as the substrate gets closer to the binding pocket, it's going to start changing its orientation into that transition state and make that a more likely reaction. So this becomes largely an enthalpy, change in enthalpy type of reaction. So we're going to look at an imaginary stickase. And I just know one of these, I haven't done it yet, but one of these years I'm going to break this ruler up here. Okay, so here's what we've got. Actually, let me go down. Got my slides in the wrong order here. Okay, so suppose our enzyme bound our substrate perfectly. So we've got our stickase, and here's our stick that we want to break. but it just fits in there nice and even. So, hey, perfect, awesome, fits in there perfectly. We're not gonna break it. What we're gonna see happen with our free energy diagram, it's gonna take a little bit of energy to get it to bind, but then this is really stable in here, and we still have this huge delta G to overcome to actually break it. So we're not gonna see a lot of reaction occurring this way. So our enzymes don't bind our substrate. What they do is they bind our, oh, I did get through here. We want to break this. Sorry, let's just skip this. So here's our state. Here's our bad catalyst, bad enzyme. What we actually want to have happen is have our enzyme bind the transition state. So instead of binding the stick, it binds the bent stick, right? And the more we bend it, the more likely it is to bind, okay? And the more we bend it, the more likely it is to break and then get released, so we make our product. So this is what we see here. Here we bind our substrate, still some binding energy here, and then we actually break it, but now we've lowered this activation energy because our stick is already significantly bent, so it's really easy to break it at this point, okay? And then once it's broken, we release it, and that's basically the way it works. Okay. Yes?
I'm sorry, I thought you said this, but ES stands for
the enzyme substrate complex. Yeah. So basically, we can have E by itself. So with nothing bound, that would just be our enzyme. We can have substrate by itself. We're going to see more of this in a little more detail here. And then when the two are together, we have an enzyme substrate complex.
and that's like kind of like a happy or like it's more stable.
Yes. Yeah. Yeah. So basically, you know, these two match up, so they want to bind. It takes a little bit of energy to get it in there, but once it gets in there, it's fairly happy in there. Okay. But yes, yeah, yeah. So we're binding that transition state. So we're not, you know, it's not like this would be perfect. You'd see this, you know, this would be really stable in here. This would be like a permanent interaction. In this case, this is not comfortable. It's like our seats at McDonald's where like, oh, they're comfortable at first, but then they get a little uncomfortable because they want you to move on, our industrial engineering here. So this is comfortable, and then we'll either get up and leave or, you know, break our back. I don't know, that's probably a terrible analogy there. Okay, all right.
So what do you do with the, okay. You break it because you don't need it no more?
So in this case, this is just an example of our stick ace is just something that wants to break sticks. So yeah. So this will just be our rough analogy for what's going to happen with our substrate being converted into products. In this case, we're breaking sticks for firewood because the heat's not on right now. But we'll see, like when we convert, you know, maybe our phenylalanine into, you know, hydroxyphenylalanine. You know, we want to do that. So we're going to have that kind of halfway in there. Our phenylalanine would bind, but it'll be maneuvered into position where it's easier to hydroxylate it. And then once it's hydroxylated, it's like, oh, I'm done. You know, kick it out. Okay. All right. Okay. All that good? Yeah, sweet. All right. So I think just to give you guys a break, I'm going to tell you why I will never be asked to, you know, be involved in freshman orientation. So I had a friend on social media post this, you know, motivational / inspirational thing today about, you know, I'm sure that this really happened, but you know, it's not about bringing a bunch of freshmen onto campus. They gave them all a balloon that was blown up, and a marker. You write your name on the balloon and throw it all into like a big room or something like that. And they said, all right, go in, find your balloon, and like nobody could find it. Okay. So then they said, all right, everybody go in and grab a balloon and then find the person whose name is on it. And in 5 minutes, everybody's had their own balloons back and everybody had new friends and everybody clapped, right? So, and they said, you know, and this is like happiness. You can't focus on finding your own happiness or you'll always be sad. But if you focus on giving other people happiness, then yeah, it'll be good. So my thought was, they're doing it wrong. You know, how do you find your happiness? You take a pen, pick up a balloon, like, oh, it's not mine, pop it, yeah, pop it. And eventually you find yours. It's a lot easier. So the key is you have to destroy everyone else's happiness so that you can find your own. It's probably not the message that they'd want there too, but that would be an efficient way. Yeah. And of course, what happens if someone else pops your balloon? You're like, oh, well, happiness was just an illusion anyways. Nothing but, you know, vacant air filling up a hollow shell. So I don't know, it's probably a little too nihilistic for welcoming freshmen to class, right? Dr. Ebert makes our exams? Okay. All right. So, I don't know. That's my sense of humor there too. So, all right. So, not too bad, right? You know, our enzymes are going to facilitate the catalytic reaction by binding the transition state. Okay, so how does this happen? All right, we're gonna zip through a bunch of this because, you know, it's too much detail for this level. This is one of the advantages and disadvantages of Lehninger. It's going to go into excruciating detail about the steps in acid-base catalysis. I would like you guys to kind of like follow through because I remember from my advanced organic class I took in grad school, the secret to figuring out what the products are going to be in a reaction, follow the electrons. So there's some nice little electron charts in here. If you can follow where those electrons are going to go, you can predict the product, all right? But we're not going to go into this super detail here, but we're going to mention acid-base catalysis, covalent catalysis, and metal ion catalysis, all right, as just types of mechanisms that happen here. So our general acid-base catalysis is probably our most common. We can, in this case, we can talk about our electron donors, electron pair donors, and electron pair acceptors. All right, we can also talk about our proton donors and proton acceptors. So here we have nucleophilic attack from our carboxylic acid. This is going to be our donor, this is going to be our acceptor, and we can make this little bond here. We've now made this oxygen atom that has an extra positive charge in here. So we can kick off that proton to, you know, some base somewhere, maybe make water, get that way, or we can have, you know, another base acceptor here that takes that, and we now end up having made this product. Okay, and then, you know, since we now have this nitrogen here that has 4 bonds, so we added another proton to it as well, something like that, we might be able to kick that off. Okay, so this is our acid-base catalysis. All right, we can basically just kind of figure out where our protons and our electron pairs are going to go, and that can help guide us in seeing how the products are being made. Okay, so in general terms, we'll often see glutamate and aspartate in this because, of course, they have side chains that are carboxylic acids. Right? Lysine and arginine, side chains that are amino groups, so those will fit in there. Cysteine with that sulfhydryl group, histidine, serine, and tyrosine are often involved in acid-base catalysis. Okay. All right. Our covalent catalysis, we're just going to rearrange some bonds a little bit here too. So maybe our uncatalyzed reaction, we have this AB, and we're going to break that bond to give us A plus B. Our catalyzed reaction, we're going to have AB, it's going to interact with X here. X is going to kind of displace B, so we're going to have AX plus B. And then, you know, we hydrolyze this bond and kick off X. So we have to have this nice nucleophilic group on our enzyme that can become a good leaving group once it's displaced the other one in here. Often this is going to be a serine, thylene, amine group, or carboxylic acid. Okay. Our metal ion catalysis, again, we're going to have, again, this is why we often have minerals that are specifically required in our diets. You know, we got to have our iron, got to have our copper, our vanadium, whatever. We have a metal ion that is bound to the enzyme. So there's our cofactor, metal ion cofactor. Remember back from chapter 3? It's going to interact with the substrate, and it can stabilize negative charges because we have this big honking positive metal ion in there. And of course, it can participate in redox reactions like we saw a little bit with our iron and the hemoglobin. All right. So if we look at an example, maybe we'll have chymotrypsin in here. We're going to digest basically chymotrypsin if you eat some nice yummy food, because I'm getting hungry because it's almost lunchtime. You know, we're going to chop up some proteins, and it's going to recognize a particular sequence after this tyrosine, right? And it's going to recognize the sequence, and it's going to cleave this bond here. Okay. So basically, one of the neat things is this tyrosine will fit into the cleft, and then we can have the activity go on there. So chymotrypsin uses pretty much all those mechanisms we just talked about. Oh Lord. So this is why I'm going to go fast. I'm not going to make you guys, you know, tell me how chymotrypsin cuts, but just for illustrative purposes, all right, we have this little oxani on the hole in here in this hydrophobic pocket, all right, this little section here. So this is kind of our binding site, a nice little cartoon of our binding site. And here we have this polypeptide, and bingo, here we have this side chain that fits into the hydrophobic pocket here we have our anion hole. We've got a nice little glycine sitting there that'll allow it to interact. All right. So it positions the peptide in, it's like basically a little pause right here and says this is where we're gonna get it. All right. So once this happens, we now have this serine over here. Okay, that is stabilized with this histidine. Okay, this histidine is going to pull off the proton, and then the serine oxygen is going to attack that carboxylic acid carbon. All right, and it's going to cause this to flip up this way. All right, so now we have this negatively charged oxygen down here. Okay, we've got this kind of bound in here. All right, so we got this nice little binding here. Well, now we can release this and basically make this cut. Okay, so when we make this cut, oh, that's terrible, it's so small. All right, so we make this cut in here as our carbon-nitrogen bond, our peptide bond. It's going to come up here and interact to bind to this hydrogen. We can release that first product. We've made our cut. We have this acyl enzyme intermediate that's now covalently bound to that serine. So we can bring in water to hydrolyze that serine-acyl bond. Okay, so the water comes in there. Again, it gets stabilized by that histidine, and this oxygen can attack that, jump these back up this way, and we can release that. So we've got this kind of stabilized in here like so. Yeah, oh, sorry, going this way. All right, and then now we've regenerated this, and we can release this out, let it go, and bring in another section of protein to cleave it together there. Okay, so product dissociation. All right. So we have a bunch of acid-base catalysis and a bunch of covalent catalysis in there. And again, this is fairly typical. Our enzyme is actually making a covalent bond with our substrate at one point. But then, when the water comes in and hydrolyzes that, we regenerate our chymotrypsin so that we can release our products, our cleaved polypeptide, and repeat the process again. Okay. So if we look at like some antibiotics, a way they'll work is they'll punch holes in the cell walls of bacteria. So a peptidoglycan is a polysaccharide in many of the cell walls in there. So if you can cleave that cell wall, you can lyse the bacteria. So lysozyme is a nice little antibacterial agent, and it will recognize those peptidoglycans and cleave them, again, using acid-base catalysis and covalent catalysis. All right, I'm not going to go through that because I'm not going to ask you about that. There is a nice way to kind of look at this, because understanding the specific mechanism can help you figure out, the way that it works, whether it's SN1 or SN2. In the case of peptidoglycan cleavage, it's 2 successive SN2 steps. I'm just going to assume that everybody knows their organic chemistry, right? So you could figure out, is it an SN1 mechanism? Is it an SN2 mechanism? But you could look at your products and see how it comes out that way and figure it out. All right. Okay. But like I say, I'm not going to ask you about peptidoglycan. I'm not going to ask you about the specifics of, say, enzyme catalysis, of acid-base catalysis or covalent catalysis or metal ion catalysis. I want you to know that those exist, and then maybe, be able to look at something and say, oh, okay, draw where the arrows are going or something like that. So I find, these types of pictures useful, because you can see, okay, here's our lone pair attacking this way, so we're going to make this double bond. Okay, so we got this, double bond intermediate in here, and we've lost this. Basically this lone pair is going to come up here and attack that way. So, if you can follow that electron pair movement and those bonds breaking and forming, I'm happy. Okay. Sound good? Yeah. All right. So we're going to introduce this today, and maybe finish it on Thursday, but probably not. We'll definitely finish it up next Tuesday. You're like, hey, it's only half a chapter. How long could it take? There's a lot condensed into these last 30 slides or so, okay? So we know that our enzymes are going to bind substrates and they're going to catalyze a reaction. So our enzyme kinetics are going to be the rate at which this occurs, okay? So the rate is affected by the particular enzyme. Some enzymes are better than others. It's affected by the substrate. Some are more prone to being converted into product than others. We can have various effectors. We might call these regulatory molecules, okay? We might call these inhibitors or promoters as well too, because there's a couple different ways that can happen. So various effectors, okay? And then of course temperature as well, right? So if we change the temperature, typically we'll see a reaction speed up. Not always, but most of the time. All right. Here are awesome role models for this, Michaelis and Minton. For the longest time, it's really interesting for me because you talk about women in science has been an incredibly successful program. I can remember starting school and I was a physics major and I dated all the women in my program. There was one. Okay, so it's not like I'm really cool or anything. It's just I dated the only female physics major. So Minton is kind of held up as, oh look, women in science. The women in science program started somewhere in the 90s, I believe, late 90s. Started developing a network of women to kind of mentor and serve as role models at all levels, whether it was middle school students, stay interested in science. Same with high school students, college students, keep interested in science. Early career professionals as well too. Okay, I can remember the first woman in my biochem department, and she was just kind of like the first person there, and very, very kind of meek. The second woman in the department was brought in this chair. She happened to be my PI, and if we go, I'm gonna make an old reference here, old Saturday Night Live. Look up Janet Reno crashing through walls like the Kool-Aid Man. That was my PI. She was a force to be reckoned with, and people were afraid of her. She was actually really nice, but she just came across as being very gruff. But again, all this kind of snowballed into encouraging women to stay in the sciences, to the point now where like biology is something like 60% women. If you look at say like physics and computer science, they're still mostly men, but women are like about a third in there. So all this kind of tails back to, hey, look, we got Maud Minton, all right, in biochemistry, all right. So what's my point there? There are avenues for success. There are other categories that we could probably focus on that should use the model of women in science to keep people in the sciences. All right. My little time waster there to give you a second. Why should we study enzyme kinetics? This is where I'm expecting you guys to go, we shouldn't. Let's just skip it. We'll move on to chapter 7. It'll be fine. Why do we need this? Well, so we can look at this quantitative description of biocatalysis. We can figure out how much, how fast, all that sort of stuff. We can figure out the binding order of various substrates, okay, that can play a role in, say, medicine development. We can figure out the specific catalysis methods, what acid is interacting with what base, what covalent bonds are being made, and this can help us tailor treatments for diseases. We can understand the catalytic mechanism the same way. We can find effective inhibitors. Our inhibitors are going to play huge roles. So for example, if you ever had a headache, maybe you've taken ibuprofen or aspirin or acetaminophen, it's like that's an inhibitor for the pain response, right? And of course, we can understand the regulation of activity so that we can build super people or just understand it to do other stuff. All right, so that we can, helpful in regenerative medicine. How do we stimulate, a faulty nerve pathway to regrow? All right, so how do we actually figure out our enzyme kinetics equations? So we're going to start with a model, okay? And we're going to have our Michaelis-Menten kinetics. Not everything obeys Michaelis-Menten kinetics, but we're going to start with that. We're going to figure out our constraints, our assumptions, do some algebra, or maybe some graph theory if the algebra gets really hard. But we're going to start with our simplest model. Okay? We have an enzyme, we have a substrate. The two of them get together, and we have this enzyme substrate complex. Oh no, one of my arrows is off there. We'll see it in soon. So we basically have one reactant, one product, and no inhibitors involved. Okay, so this is better. This is the better equation here. Enzyme plus substrate. we get our enzyme substrate complex. You know, this is freely reversible. Our substrate can fall off without the, you know, it's kind of like, you know, the condo tour. You get a 3-day vacation and you can try and sell you a condo, right? You can go down there, you don't have to buy the condo, you still get the vacation. So, you know, you go down there, vacation, and you leave. Or you can buy a condo and, you know, whatever. I was talking about that to me yesterday. Anyway, we're going to make some assumptions here. The total enzyme concentration is going to be constant. So we're not going to add or subtract enzymes in there. That does happen over a time scale, but we're going to assume it's constant. So our total enzyme is going to be our free enzyme plus our enzyme that has substrate bound. Okay, so total enzyme concentration is a concentration of free enzyme plus substrate bound enzyme. We're also going to assume that our substrate total, okay, is roughly equal to our substrate concentration. We're not going to try and, just like we saw with the kinetics on the previous one where we, you know, did the algebra there, the quadratic equation, we could just estimate and we'd still be within like 1%, less than 1% error. We're going to do the same thing here. We're going to assume that our substrate concentration is much, much greater than our enzyme concentration, which is true in pretty much every case biologically, okay? We're also going to assume a steady state, okay? We're going to assume that the rate of formation of the enzyme substrate complex is equal to the rate of the breakdown of the enzyme substrate complex, okay? So basically, as quickly as we're making this, it's breaking down. So that's our steady state, okay? So our observed rate, our observed velocity is going to be the change in product over time, okay? Or we could talk about this in terms of a rate constant times our concentration of ES, okay? All right. The only problem is we can't really measure the ES concentration very easily. There's like a very few limited ones we can do that, but we can't. So we end up substituting in here, all right? So we'll take a look at this algebra on Thursday, but whoops, wrong way. But we're gonna start with this equation. We're gonna substitute in for ES based on what we have here, okay? So we'll end up with our velocity is gonna equal this K cat times the total enzyme times ES over Km. So if we assume that everything's firing as fast as it can, that'll be our maximum velocity. We'll say K times the total enzyme concentration is Vmax, okay? All right. Our K cat is our turnover number. How fast, how many substrate molecules can one enzyme convert each second, okay? And our Michaelis constant is how strongly does our enzyme bind the substrate. So those are the two factors that are gonna affect it. I think of K-Cat like that classic I Love Lucy episode where she's in the chocolate factory and has to put the chocolates in the box or something like that. And then the conveyor belt goes faster and faster and she's like stuffed in her mouth and her hat and just so fast. Our K-Cat is the maximum number we can convert. Our Km, how well can we pick it up, okay? All right. So during the steady state, the Vmax occurs when all the enzyme is in the ES complex, okay? So that's basically what we got going on here. Microscopically, well, this is really going to depend on the details of the mechanism, but we'll see. All right, so we can kind of take all this and plot it. All right, so our velocity is Vmax times substrate over Km plus substrate. The neat thing about this is the velocity of our reaction, our rate, only depends on the concentration of the substrate, not the concentration of the enzyme, okay? Just the concentration of the substrate, okay? So if we were to do an experiment here to make, we would mix our enzyme with our substrate. We could record the rate of the substrate disappearance, or maybe the product appearance, depends on which one has the best spectroscopic properties for us to record. And then we can plot our initial velocity versus the substrate concentration, all right? And then we can plot the change of the substrate concentration and repeat. So essentially here's what we have. This is our substrate over time. We monitor its concentration by absorbance. We'll see as it's being converted, this is going to go down, all right? So our time over mixing here. We take a look at our slope here. We start plotting product concentration versus time. We're going to see our product concentration is going to increase. If we have, you know, different substrate concentrations in here, you know, we're going to max out at different, you know, you can see we're maxing out at the same time, but we're just making more product there, okay? So our ideal rate, okay, is, you know, velocity equals Vmax times S over Km plus S, okay? We can see deviations in this because of, you know, limitations of our measurements. Maybe we just can't detect our product or substrate very well. We can have inhibitors present. You know, so basically inhibitors are everywhere. It's like RNAs is, all right. But if we plot this, we end up plotting substrate concentration versus our velocity. Okay, we'll get this hyperbolic graph that approaches a theoretical maximum velocity. Okay, this is our asymptote. We're never actually, statistically, we're never actually going to have all the enzyme sites occupied, but we can get close to that. Okay, so that's our asymptote up there. What gets very interesting is when our substrate concentration is equal to our Km, we're at half Vmax. So if I go to my equation, all right, that's not what I wanted. Nope, let me go back up that way. All right, so when the substrate concentration equals Km, all right, we'll essentially have, you know, 1/2 Vmax. You can see the math in there. So that's kind of a neat thing we can see here. Okay, we might not know this, but we can often find our 1/2 Vmax to figure out our Km. So if we have an unknown enzyme, we can figure out its Michaelis constant by plotting its interaction with its substrate. Okay. All right. We can look at saturation kinetics. Okay. At very high substrate concentration, our velocity is not proportional there. We just have this, you know, very quick spike. Our initial thing is like, let's go. You know, all the football players run on the field at the start of the game. It's like, woo, we're ready to go. That's our initial velocity there. All right. So we essentially have something that looks like this, but, you know, it's just our initial curve. We're not going to talk about that too much. We're going to look into our kinetic parameters. Okay. Our nonlinear Michaelis-Menten plot is, you know, useful, but a lot of times, you know, we can get better data by making this linear. So we can use this double reciprocal, okay? Instead of plotting substrate concentration versus a velocity, we'll plot 1/over velocity and 1/over substrate concentration, okay? And that will give us a straight line, okay? And this is known as a Lineweaver-Burke plot. And get used to these because we're gonna see a lot of these. And as we change stuff, we're gonna see how these change as well, okay? So the neat thing about this is that our y-intercept is 1/over Vmax, our x-intercept is -1/over Km, and our slope is basically Km over Vmax. But by making a Lineweaver-Burke plot, we can determine both Km and the Vmax, okay? Much better than our hyperbolic curve, where we have this asymptote up here, but we can't quite get to it. And then we don't exactly know what our 1/2 Vmax is. With a Lineweaver-Burke plot, our intercepts are those two values. And that's why that's useful. Okay. All right. I'll just peek ahead at this. Our Kcat over Km is our total enzyme efficiency. It's basically, we can look at both Kcat and Km and how they affect the efficiency of the overall enzyme. If you look at acetylcholinesterase, it binds acetylcholine. The Kcat is 1.4 × 10 to the 4th. The Km is 9 × 10 to the -5th. So that's, you know, fairly average in there. You divide these two and you get 1.6 × 10 to the 8th. That is a very fast, very efficient enzyme. Okay? Essentially, we are approaching the diffusion limit. Molecules can only diffuse at a rate of about 10 to the 8th, 10 to the 9th inverse moles per second. Okay, so that we're basically, acetylcholinesterase is so efficient that as fast as you can make that acetylcholine, it'll suck it up and convert it into product. Okay? We can see catalase in here, which is going to bind our peroxide. So basically, our peroxidases are also close to that limit. You know, so we want to scavenge that stuff in there too. If we look, that's the best one there. Yeah. Okay, so Kcat over Km can just basically tell us how fast we can, how efficient our enzyme is overall. And you can see that there's a couple different ones here. Like for example, the Km of catalase is really low, but its turnover number is really high. So when you divide these, you still get a very high efficiency overall. You might have others that have a really strong specificity, but a much lower Kcat. But you still, when you divide those two, that efficiency is still very high. All right, that's where we'll stop for today. When we come back, we'll take a look at how we actually derived this equation. Right, so we'll go through that. That might be a good thing to try and do on your own, and then we'll do that on Thursday. And then we'll start talking about what happens if we have, multiple substrates, and then what happens if we have inhibitors, and what happens, how do we figure out these mechanisms, okay? All right, so enjoy. Yeah, good times. Yes?
So the homework's gonna be given right in the test.
Yes. Yes, yeah, yep, yeah. So
This is
not this Thursday, but next Thursday, yeah, yep. 5. So it takes a while to get through the first 2 because they're both 3 chapters, but then they start coming a little bit faster. Like I say, exam number 2 is probably the toughest exam historically. Exam number 3 historically is like 20 points better. So you do get a break after this, but I'm throwing you into the crucible and grinding you up to see what you can do here. All right. Okay. All right, I will see all you guys later.